Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really glad you're here today. We'll be talking about inhaled JAK inhibitors and lessons about AO and lung delivery with Nikki and Neil today. Welcome and good morning and good evening to you, Neil.
Hi, thanks for having us.
Yeah, absolutely. My pleasure. And now, without further ado, I want to introduce our guests, Nikki and Neil. So let's start with Nikki. Nikki
Hi.
joined Merck as the Director of Discovery Chemistry in 2025, and prior to her role at Merck, Nikki held roles of increasing responsibility in the GSK MedChem organization. Starting at 2014, she spent 5 years working on respiratory diseases before moving into the Discovery High Throughput Chemistry group. And Nikki most recently was the leader of the oligonucleotide chemistry innovation team within GSK's modality platform, and she was responsible for both the US as well as the UK. She started her career as a medicinal chemist at Lexicon Pharmaceuticals in Princeton, New Jersey, where she most notably was a team member and inventor of sotagliflozin, also known as INPEFA, and that is a dual SGLT1 or 2 inhibitor for the treatment of type 1 diabetes and heart failure. Nikki earned her PhD from Caltech with Dave McMillan on development of application of novel organocatalytic strategies. So Nobel Prize winning work right there. Nikki is very well known for her extensive commitment to the chemistry community, and many of you might recognize her as the organizer of the anticipated first-time disclosure sessions at the ACS national meetings, and she chaired that session from 2020 to 2024. And in 2024, she was actually the elected vice chair of the ACS Medi division and currently serves as the program chair for 2025 and 2026. Nikki has also been recognized as a 2022 ACS WCC Rising Star and a 2017 Edison Patent Awardee. Welcome, Nikki. We're really glad to have you.
Thank you. Thank you for having us.
Absolutely. And yeah, let's also introduce Neil, and hopefully I'm gonna do a good job on Neil's last name. So Neil Fazakerley, was that okay? Is
Sounds good.
Exactly. Close enough.
okay. Is the global lead of GSK's modality platform. Neil also joined GSK in 2014, same year as Nikki, and started working on respiratory diseases as well before moving into GSK's Discovery High Throughput Chemistry group, where he met Nikki, I believe. Prior to his role at GSK, Neil worked as a process chemist at AstraZeneca, where he was actually a part of the team developing the manufacturing route of osimertinib, also known as Tagrisso. That's AZ's blockbuster EGFR inhibitor for the treatment of non-small cell lung cancer. Neil received his master's in chemistry from the University of Manchester, where a one-year placement at AstraZeneca confirmed his passion for drug discovery. And he stayed in Manchester and earned his PhD with David Proctor on the total synthesis of pleuromucilin, utilizing the Proctor samarium iodide radical cascade chemistry. So welcome to the show, Neil and Nikki.
Thank you very much.
All right. In the good old Drug Hunter tradition, we're starting this with a few kind of, warming up questions. So Neil and Nikki, what inspired you to start a career in drug discovery?
Neil, do you want me to go first on this one? We'll alternate.
Yeah,
All right.
go
So
for it.
I became a chemist in drug discovery because I wanted to be a doctor growing up. And here, fun fact, I don't tolerate the sight of blood very well. And so instead of being the doctor, I wanted to make the drugs to help people. And that chemistry desire actually came from attending a chemistry camp when I was early in high school at Muhlenberg College, which is up the road here. And so, kind of all things lead together. And when I went out for jobs, I applied to be in process or medchem, and I took a role at Lexicon in medchem. And different problems, different solutions, and I don't regret. And I'm very fortunate to have actually landed in a medchem org.
How about you, Neil?
Yeah, so my passion really did start at high school as well. So I had the chance to do a work placement at AstraZeneca in high school for just a couple of weeks. I think as part of that placement, lots of high school kids will have made and crystallized paracetamol as part of their, spending time on a work placement. So I did that. I took the photo down the microscope, and that really propelled my interest in chemistry, my desire to make molecules. I've always enjoyed making stuff, and really believe, and I'm really excited to have a career where I get to affect human health. And that
Awesome.
really drives me.
Yeah, follow-up question on that. So what is your favorite molecule and why?
Well, so I always still say plurimutalin, only because it still gives me nightmares. And when I joined GSK, I discovered that they had half a kilo of the material I'd spent 3.5 years making 3 mgs of. So it's
Oh my goodness.
Yeah, that's amazing.
I say that the compound of my nightmares is cyclophane, but no way is that my favorite molecule. I think the obvious answer is the molecule that we worked on, and working on sotagliflozin, SGLT1/2 inhibitor. But the reason it was really fun is that it came down to that program, whereas we're going to make one more molecule, and if this doesn't get us where we need to go, this program's done. And that one molecule worked, and it was a great effort. I had just joined at the time, so I was really like absorbing everything, and it was just a really fun program, fast paced, a lot of energy, a lot of excitement, and I just never forget those days. So, and it ended up being a drug on the market, and I can still draw it in my sleep with its stereochemistry. So, I would say that's it.
Yeah, that's amazing. I think you both can say you have an approved drug on the market. Like how many chemists can say that? That's really amazing. So what are you both most excited about in drug discovery today?
This is my turn to go first. I think seeing how we are able to address what we used to call the undruggable, and being able to say, as you're trained in synthetic chemist, you are not trained anymore to make a molecule below molecular weight of 500, right? As a synthetic chemist, you now have the power to make things that you can isolate and put in a vial, but they don't have to be your traditional small molecule. And so if you're looking at a heterobifunctional, or you're looking at an oligonucleotide or an ADC, your training, your skill set to put bonds together is just opening up this whole area where we can have an impact on people and also personally have success as things going forward. And I think seeing this evolve, it's been like a renaissance, and feels like in the past 10 years. And it's a really exciting time to be a medicinal chemist.
Yeah,
Couldn't
and
agree.
I'm going to stick with where a lot of my group is focused at the moment in high throughput and particularly nanoscale synthesis. Seeing how much data we can generate on really tiny quantities of compound that we're generating on 1536 well plates, doing that nanoscale chemistry, getting new transformations in that nanoscale chemistry that accelerates our medicinal chemistry exploration of space. That's what I'm finding really exciting at the moment.
Yeah, no, that's actually super cool. And like having the ability to screen in nanoscale as well, not only reactions, but also the biology. I find this incredibly exciting as well. So yeah, you both made it, right? You both, again, as we said, got a drug to the market, really affecting patients' lives. So, what kind of career advice would you give to people who thrive to do something like that? What kind of career advice would you give your younger self, or maybe people in the audience that start their careers in drug discovery today?
Well, I guess I cheated because I joined process chemistry and worked at the end of the process, and so that's probably the most surefire way of getting something to the market is get in it towards the end of the process where a lot of the hard work is done. I think worry less and really get all of the data you can from every experiment you do. Think about why you're doing that experiment, and really get every insight you can from each of your experiments. It was advice that was given to me early in my career, and still now talking to folks. How are we getting every insight from what we invest our time in doing in the lab?
And I'll capitalize on something you started with, Neil. But I think what I would have told myself 15 years ago is give yourself a little bit of grace. I think knowing what you bring to the table and what you know, and knowing that Neil knows something different, I don't need to know what Neil knows, right? And every day is a learning experience, and hopefully you're learning every day. And if you ever get bored, or mundane, the day-to-day stuff, maybe it's time for you to think about what that next step is going to be. But I think one of the biggest recognitions is knowing that your leaders don't often know everything either. And so you may want to give yourself some grace, and that'll alleviate a lot of, I think, headspace and anxieties. When I started my career, it would have.
Yeah, thank you both. Yeah, last question before we get started. What do you like most about Drug Hunter?
Is this me to go first, Neil?
No, I'm going to jump in first. I really like the molecular highlights right on the homepage. Seeing those scroll through, looking at those new molecules coming forward and being disclosed is really helpful.
I'll take it personal, but I really like the flash talks because when I was unemployed for a week, I couldn't get journal articles and everything. And actually, I watched a bunch of flash talks because we had public access to those. And I think they're really educational content and give you references if you want to follow up and you get journal access back. And so you guys do a great job with that, Dennis.
Awesome. Thank you so much, Nikki. Really appreciate it. So yeah, one quick thing that I wanted to kind of show before we get started. Actually, our team at Drug Hunter, I'm showing my screen here right now, really expanded over the last year, and I want to show you a few people who we recently hired, so you're aware of that Drug Hunter is actually a knowledge platform for industry scientists from industry scientists. So we have Lou with us here, who is basically has his 1-year anniversary at Drug Hunter coming up. A veteran in the industry as well. Most recently, head of platform discovery at Chimera Therapeutics, joined us about a year ago. John Overington, we hired approximately 3 weeks ago. He has been chief data officer and chief technology officer at Exscientia before he joined us. And then, of course, he's actually known also as one of the builders of the Chembl database. So really, he's going to bring the Drug Hunter data collection to a whole new level. Matt Hess is also celebrating his 1-year anniversary with us. He was working at Novartis as well as Sage, working in molecular glue degraders, also on infectious diseases, as well as in the depression field at Sage. We also were recently joined by Theo here. He has worked in many San Diego biotechs, such as Effector, but also Bellhara Therapeutics, and joins us with a lot of experience in screening methodologies for chemoproteomics, for example. Rory has been with us for about a year now. He's worked in oncology before at Insight. Brad has about 25 years of experience here and has most prominently worked at Vertex and the cystic fibrosis correctors. We're joined by Shema. She has a PhD in pharmacology, actually. She's currently out for maternity leave, so I want to wish her well here and through the flash talks as well. We recently hired Sandrine. Sandrine is our first European hire. So she's previously worked in CNS diseases at Idorsia and joined us in the beginning of this week and is now located in Basel, again, as our first European hire. We're also joined by Chase, of course. He has more than 30 years of experience, both in the biotech industry as well as in teaching medicinal chemistry, and he joined us recently as well. And then Vinicius has a very impressive background as a computational chemist as well, and delivers a lot of the content and the data that we use here at Drug Hunter every day. And then, of course, you're all familiar with our founder and CEO, Dennis, who started Drug Hunter about 8 years ago after a very successful career in industry at Genentech and Rap Therapeutics. And with that, I will stop sharing here and I'll hand it over to Nikki and Neil for their presentation on inhaled JAK inhibitors and our lessons on AO and lung delivery. Really excited for this talk, Nikki and Neil.
All right. Thank you, guys. And as we get started, I do want to thank Neil and everybody else at GSK. With Dennis and I talked about giving this talk, I was still working at GSK. And I appreciate them letting me still be able to tell part of the story and be able to share it with Neil. And I'm very grateful for my time there. And so thank you guys for allowing me to finish up this way. All right, Neil, let's get into the next slide. And I'm going to switch you guys to a different monitor. So did you go to the next slide, Neil?
No, there we go. And we're off.
There we go. Okay, so I'm switching you to a different screen here. All right, guys. So thank you guys all for joining in. It's really fun to see everything popping up on the screen and where you all calling in from, even early in the morning out in San Diego. So Neil and I today, we're going to trade off sections of the talk and bring you through this narrative on the screen in the next 30 minutes. So I'm going to start with some basics of asthma and considerations in targeting the lung. And then Neil's going to pick up on why GSK was interested in the JAK pathway for asthma and begin the MedChem story, which, like probably every story all of us have been on, it has a really good run until it hits a challenge. And so for that, I'll take a tangent into the challenge of aldehyde oxidase, its victims, its reactivity, and then Neil is going to conclude with how aldehyde oxidase activity was mitigated. And throughout the talk, we will abbreviate aldehyde oxidase as AO. Okay. So without spending too much time on this slide, because most of you know asthma, I think it speaks to the prevalence of the condition that is a chronic inflammatory disease of the lower airways. No cure exists, but symptoms are managed, right? And those symptoms are the wheezing, coughing, shortness of breath that you might be familiar with. And that tracks back to ultimately inflammation of the airways and bronchoconstriction. So there are plenty of options on the market now for standard of care. So things like you see here, of course, going to highlight the GSK portfolio, right? But the most common being Ventolin or fluticasone. These are steroids that are often used for a first line of treatment. Fluticasone is the active ingredient in Flonase. Or we could think of things like LABAs and LAMAs, and you could take them individually or in combination. And that's great if you respond to that. But if you don't, and that's about 5 to 10% of the population that are considered severe asthmatics, the current standard of care does not suffice, and patients don't have adequate responses. And so there still is room for other things to really be able to help people that have these kind of severe asthmatic conditions. Next slide, Neil. Okay, so the field has seen obviously major advancements with the advent of biologics coming to the market. And these biologics target and control very specific inflammation pathways, and they have excellent response rates. So that's great. But I think the drawback on that is that it's really important that the patient matches the pathogenesis of that pathway. So for the example with Nucala here, which targets the IL-5 pathway, severe eosinophilic asthma patients in particular see a really excellent response rate to this. So the downside is that the pathology of asthma is quite complex, and there is no one specific pathway. So there's room for new medications to help people who cannot manage the disease with their current medications. So let's transition into inhaled lung delivery. And when you think about an inhaled approach, it sounds easier than it actually is. It sounds like, oh, I just take my drug and I aerosolize it and it gets delivered to the lungs. But really, one of the really hard problems with this area starts with the translation of your preclinical models to humans. Also, your cellular models to in vivo models is not so straightforward. And that starts with the fact that the basic anatomy of our lungs between humans and preclinical species, it's just different. So humans have 5 lobes. They have 3 on the right and 2 on the left. And since I can only see Neil and Dennis and Lou, do you guys know why there's 1 less on the left?
I don't.
We need somewhere. We need somewhere to stick our heart, Nikki.
Exactly. Our heart's got to go somewhere. So fun fact is rodents also have 5 lobes, except for they have 1 big one on the left and 4 smaller ones on the right. And dogs have 6. So as you can imagine, it will make a difference where and how much drug is deposited in the different tissues and different areas of the lungs. And so this is already a source of variability that you can see flat out. And it needs to be considered as a complicating factor in scaling and ultimately doing human dose predictions. It's more difficult than an oral compound. So let's talk about the deposition of the drugs. So for an inhaled aerosolized drug, we showed this simple inverse curve that I took from this reference that's at the bottom of the page. It's a nice reference. And it shows the hypothetical relationship between local drug concentration and your airway surface area, which basically means that the highest compound concentrations, so the smiley face on the red line, that falls closest to the inhalation point, which is your trachea. And the trachea has the lowest relative airway surface area, which is that corresponding blue line with that arrow pointed down to the bottom. So the further away you get from the inhalation point, the trachea, the surface area increases as those airways continually branch out and get smaller to maximally deliver air throughout the lung. So you might expect that with more distance and smaller roads to travel, less aerosolized compound is deposited. So for diseases like COPD and asthma that are diseases of the small airways, it's really important to try to move, if you will, turn this frown upside down, but move that frown face towards the left and get higher concentrations deeper into the lung. And the simplest way to do that comes back to physics. So when we think about inhaling a compound from an aerosolation device, like an ellipta inhaler or your dry powder inhaler, like your Ventolin inhaler, we want the particle size to be small and uniform. And a good ED50 tends to be about 10 microns. If it's too big, it won't get very far. But if it's too small, its physical properties start to be compromised and it'll actually agglomerate. So the inhaled drug delivery and engineering is a feat all in itself. And that would be like a totally different 30-minute talk to dedicate to that. So we're going to move on. So you'll not be surprised too to know that different sections of the lung have different cell types. And these have been nicely studied and they continue to be studied. But simply put, if you're targeting a disease and you want to enhance macrophage function, you need to deliver where the macrophages are. It's also not surprising, too, that different cell types have different metabolizing capacities. And sticking with macrophages, the job of macrophages is to engulf and degrade foreign bodies. So it's not surprising that it has a high metabolizing capacity. And so these differences from species anatomy to particle size to the different types of cells and their metabolic capabilities makes it a inhaled administration route very challenging. So anything that a medicinal chemist can do to take away liabilities on our end to have a cleaner compound, if you will, is desirable, since that in vitro and in vivo translation is already quite complex and difficult enough. So at this point, Neil's going to take over and tell you more about JAK and our medchem efforts.
Thanks, Nikki. So this leads us to JAK, part of the synonymous JAK-STAT signaling pathway that is essential for our immunomodulatory responses. Type 1 and 2 cytokine receptors comprise subunits that physically associate with intracellular Janus kinases, or JAKs. Alone, these receptors are inactive. When extracellular cytokines associate with the receptor, it is activated through a co-location of the JAK subunits, which leads to phosphorylation of JAK through their tyrosine kinase activity. This phosphorylation of itself triggers their enzymatic activity, in this case by creating docking sites for signaling molecules called STATs. STATs are phosphorylated by enzymatically active JAK, leading to their dimerization. Phosphorylated STAT dimers translocate into the nucleus and set off gene transcription. Unlike therapies like monoclonal antibodies that prevent their specific cytokine from binding to its respective receptor, a small molecule JAK kinase inhibitor would operate intracellularly and affect multiple signaling pathways. Most inflammatory pathways implicated in asthma involve cytokines and growth factors that signal via these receptors coupled to JAKs. JAK's role in immune responses associated with asthma are highlighted in green on this slide and have been studied in genetically modified and cellular models. As you can see, different pairings of JAKs impact different pathways, and several of these pathways are associated with on-target safety concerns, including infection risk and cytopenia. These are not necessarily nose right. There are JAK inhibitors that are market approved. This emphasizes the need to be deliberate in decisions made in the risk-benefit profiles because the health of patients is paramount when new medicines are brought forward. One of the things you might be thinking is to target selective JAK inhibitors, which is a viable path depending on the disease indication. However, selective JAK inhibition has been observed to lead to incomplete responses and thus leads to increasing the dose, which has concerns for tolerability and safety both on and off target. Another way is to go for topical administration, like dermal or inhaled, and designing the compound to have high exposure at the site of action but low exposure systemically, effectively giving an increased therapeutic index. This approach is evidenced for dermatology indications like atopic dermatitis and for inhaled administration. An example of this is the reportedly selective JAK1 inhibitor from AZ that is currently in phase 2. In our JAK program, we felt that developing a pan-JAK inhibitor compound would preserve the broadest pharmacology in the lung possible, but if designed to have high systemic clearance, there would be an adequate therapeutic index for a medicine. An internal focused HTS identified aminoquinazolines like compound 1 as a pan-JAK inhibitor of moderate potency. At GSK, we routinely consider binding and inhibition in terms of PIC50, the negative log of the concentration at which 50% inhibition is observed. So 1 is 80 nanomolar at JAK1 and 250 nanomolar at JAK2. Considering the aminoquinazoline motif as the hinge binder, an essential interaction for kinase activity, SAR was conducted at the 8 position, where it was found amino linkers like 4 and 5 were preferred. Crystallography of this compound confirmed the expected binding mode, with the aminoquinazoline nitrogens making a 2-point connection to the hinge region. This is the region that binds the heterocycline ATP that you are competing physiologically with. The piperidine projects into the ribose pocket, where we see there is some space. But what we're not seeing is interaction with the back pocket, sometimes called the selectivity pocket. The gatekeeper residue is this methionine, and there's a water network anchored to a glutamic acid back there, making it a prime area for medicinal chemists to target for potency and kinase selectivity. In order to continue to grow the molecule, the ligand efficiency of this set of compounds was compared, and it turns out that compounds 3 and the most potent one, 4, have similar ligand efficiencies. But with compound 3 having a lower molecular weight, this scaffold was chosen to continue to progress. With the plan to explore into the back pocket, which is highlighted in yellow. So the pyrosol region is highlighted in green, and it was previously observed that the phenol unit was effective in disrupting this water network through binding to this glutamic acid. And indeed, compound 6 jumps 1,000-fold in potency into the subnanomolar range at 0.5 nanomolar at JAK1. But looking at this, you're not surprised that the solubility drops significantly as a result of this aromatic addition. Good news, though, is that the desired interaction to the back pocket glutamic acid residue was observed in a crystal structure, confirming the hypothesis for increased potency of this unit. Looking closer to the hinge region, it does not appear that the pyrosol is serving much binding function, and it's just heading out to solvent front, which we usually think about for improving properties. But in this case, removal of it increases the solubility, and it did just increase that solubility, but with 100-fold loss in potency. However, in the bottom, there's no change in the binding site. So despite the potency difference, the loss in molecular weight gives us ligand efficiencies in the same range. So compound 7 was used to continue to optimize this series. Optimization in that back pocket region led the team to compound 8, which retained solubility but showed about a 6 to 10-fold increase in potency. It's believed that the attenuated pKa from the phenol at about 10 to the fluorophenol at about 8.5 enhanced the strength of this interaction. In addition, with this side-on view, we appear to just be filling the space better. The torsion angle observed in the crystal structure also matches closely the torsion angle of the calculated ground state conformation, meaning that pre-organizing this conformation through the orthoethyl is giving this molecule an entropic benefit for binding, thus increasing the potency. Now, while the kinetic solubility from DMSO into buffer is good, we also look at thermodynamic solubility from solid in more physiologically relevant systems. In this case, solubility in simulated lung fluid, or SLF. This is a mixture of albumins and other proteins, antioxidants and salts, with a buffer at around pH 7. As is commonly observed, the solubility of these molecules went from moderate to bad, which for inhaled drug is a significant risk for administration. In soluble compounds, risk causing macrophage toxicity, Thus, to address solubility concerns, the team moved to add polar groups to the 2 and 8 positions, where there was space to expand. Through contemporaneous studies, I'll show the 8 positions first, and amino alcohol additions were proven to be of the most interest. Biochemical potency was improved, and now on this slide, we're looking at activity in cell-based assays. In this case, HLF is human lung fibroblasts that were stimulated by IL-13, and inhibition was measured by eotaxin release. Cell-based activity is still observed both in this HLF model and in an IL-2 stimulated PBMC assay, although not shown on the slide. With these substitutions, I'm only showing 3, the SLF solubility is improved, and we see a desirable high clearance in hepatocytes, most likely owing to the secondary metabolism of the phenol. Remember, for the profile we want, low systemic exposure, high clearance is a good thing. So compound 11 was an early tool compound for in vivo studies, and intranasal mouse PK showed the desired difference between lung retention and systemic exposure. And while the curves look pleasing, it would be better if the y-axis was a log unit or so higher for the concentrations observed. We're not able to cover a desired IC90 profile for more than 30 minutes. Additionally, you can see from the dashed line, we were well below IC90 in unbound concentrations. And really want to highlight here the importance of considering protein binding and unbound concentration, which is well documented in the literature. So an okay start, but lung retention was needed for inhaled administration. And there are 3 known ways to increase lung retention. Number 1, solubility, relying on dissolution rate, but I've already described that that's not a great solution. We can decrease the permeability, so the compound doesn't pass through the lung tissue so quickly, or we can increase the volume and the lung tissue distribution. And a known way to do this is to increase the basicity of the compound. So cutting some SAR to the chase for time, this was done, but instead of the desired effect, the lung exposure looks even worse, and all the properties and potencies were similar, as you might expect with this group heading out into the ribose pocket. As part of our compound profiling assays, we do survey common metabolic pathways like CYPs and microsomal and hepatocyte stability. We also do aldehyde oxidase, particularly because many drug structures have heterocycles that might be susceptible to AO. Indeed, when compound 11 was incubated in human and mouse liver cytosols, where AO is located, without an AO inhibitor, which is the blue line, it gets consumed very quickly. Adding a known aldehyde oxidase inhibitor shows that metabolism, and in this case, it was confirmed that compound 13 was the metabolite observed. And in its own right, it's a weakly active pan-JAK inhibitor. So aldehyde oxidase isn't one that we hear about as much as counterparts like P450s or UGTs. AO poses a significant in vitro, in vivo translation risk. So we thought it would be good to do a review of some basic facts for AO. First, while other species like rodents have many isoforms of aldehyde oxidase, humans only have one. It's expressed largely in the liver and adrenal glands, but recent proteomic work does find aldehyde oxidase 1 expression in human lung. Just to make sure, compound 11 was incubated in lung tissue homogenate of both human and mice, and the same oxidative metabolism was observed in both, leaving the team with a translation risk to human. But how do you think about the AO differences between species? So to learn more about this, I'm going to hand back to Nikki to discuss the challenges and opportunities of AO metabolism.
All right. Thanks, Neil. Am I coming through okay? Yeah,
I hear
Dennis
you.
is nodding. All right, thank you. Okay, so some of the challenges with AO activity is, one is that there's less of an understanding of its biology compared to something that's much more commonly studied, like P450s. So therefore, predicting and scaling human PK with this liability is difficult. And I really do want to emphasize what Neil already alluded to, is that while humans have one isoform, rats, dogs, other your preclinical species have others, and they are distributed sometimes much more ubiquitously than they are in a human. The SAR of CYPs, which is obviously our most common metabolizing enzyme, the SAR of CYPs and aldehyde oxidase run counter to each other. So, for example, if you add nitrogens to your heterocycle to attenuate CYP oxidation, you might now find yourself with AO liabilities. And these types of nitrogenous heterocycles are so common in drugs that it can be hard to fully remove AO activity. All right, so in addition, as we alluded to in the beginning of the talk, the understanding of lung metabolism and expression of these enzymes is not well characterized, but there has been significant advancements made over the past decade. And I really draw your attention, if you're interested, to this review from UCB down at the bottom. And I took this figure from there. And it listed metabolizing enzymes found in human lung tissue, and they were found from genotyping. So here's aldehyde oxidase in the green circle. And what I want you to take away from this fairly busy slide is that one, aldehyde oxidase is not considered the most relevant. Those are in red. And it's also not considered the most established. In other words, there's not the most known about it. So it's there, but how to give relative weight to its metabolic capacity when you observe this liability, it's not well understood. But what we do know is its structure, and we know that it's a cytosolic butterfly-shaped protein, which is about 150 kilodaltons in weight, and it consists of 3 domains. So one with iron clusters, they're in green, that's for redox cycling, which is right next to the FAD domain, which is in blue. And then the one we're going to talk more about, the domain where the molybdenum cofactor is located. It's affectionately called MoCo, and that's what's listed on the slides. And so what's reported from crystal structures, and we show a zoom in of it on the right, is that within that MoCo binding site, the blue arrow is pointing to the molybdenum. You can see that there's a nice kind of surface and ability to dock a heterocycle. So that simple, I think it's a quinoline there, is shown in green. So AO has 4 distinct classes of chemical reactivity. And the most well-known by far is heterocycle oxidation that occurs through a molybdenum 4,6 redox cycle. So molybdenum 4, which is at the bottom of that cycle there, if you will, is activated in a 2-electron process to a molybdenum 6 intermediate, where that nucleophilic OH that's attached to the molybdenum is likely further activated through the neighboring glutamine and lysine that attacks the heterocycle. It's postulated that concerted mechanism does a hydride transfer, and the cycle is turned over by exogenous water. And there are some deuterium labeling studies that confirm that role of exogenous water in turning this over. So with this kind of CH activation-like activity at play, it's not surprising, to people on this call that heterocycles that would be more at risk are those that are more electron poor and those that have more nitrogens. And so to mitigate that, you can decrease your nitrogen count or increase the electron density of your heterocycle. Also, 6-membered rings tend to be more susceptible than 5-membered rings. And many of these images are taken, again, from another excellent review on this topic that was 5 years ago in JMedChem. And I'm showing some of the list of examples of heterocycles that can be susceptible to aldehyde oxidase activity, but this reference has a much more encompassing list of that. And I encourage you to go there. So here's a nice example from Vertex of how they thoroughly studied VX509, which ironically is a selective JAK3 inhibitor. And this compound was found to have an aldehyde oxidase liability in the clinic. So despite being deemed a low drug-drug interaction or DDI risk based on its preclinical profiling, in the clinic, when they administered it with midazolam, which is a CYP3A4 substrate, they found a significant increase in the area of the curve for midazolam. Means that VX509 is having an impact on CYP metabolism, and it's also a risk for human DDI. This paper from Vertex, that's in that gray box there, does a really nice job characterizing what's ultimately a TDI, and it comes from this particular metabolite that's shown on the top right there, and they identified that as the product of aldehyde oxidase metabolism. And just to remind the audience that preclinical profiling with microsomes will not capture aldehyde oxidase activity, as it's a cytosolic enzyme. So you do need hepatocytes for that. And what many of us also know is that there is a lot of batch-to-batch and donor variability that can happen with hepatocytes. This paper actually goes into their experience was that. And so that's just a reminder that we always need to run the right control experiments to accommodate that batch-to-batch and donor-to-donor variability. So significant kinetic studies were run. I'm not going to be the expert to present those, but if that's your jam, absolutely look into that paper. And they ultimately predict the human DDI and carry forward. But this compound was ultimately discontinued for safety signals. So as per its namesake, a second mechanism for reactivity of aldehyde oxidase is that it does oxidation of aldehydes. Shocking, right? And while aldehydes is not usually in your final drug, it can be produced through other metabolic pathways. And so I think a good example of this along the way kind of reactivity is imricoxib, which is a now discontinued COX-2 inhibitor. So the methyl group in that phenyl ring underwent 2 P450 oxidations to the aldehyde. And once the aldehyde was formed, it was aldehyde oxidase that was the major factor in oxidizing it further up to the carboxylic acid. And it was an unexpected finding in the clinic that they had to deal with. And ultimately, said this compound is not on the market. The next class of reactivity is actually counter to its name, and that aldehyde oxidase can do reductions. As one example of taking the nitro down to an amine, as is what's seen with dantrolene in the gray box. And for this compound, unfortunately, formation of that aniline led to subsequent transformations and metabolic events that cause safety signals. So you might be thinking like, hey, wait, there's like one enzyme that does oxidation and reduction, and others have asked about that mechanism too. And it is postulated, and may not be, surprising, but it is postulated to occur at 2 distinct binding sites on the enzyme, and reduction is likely occurring at the FAD binding site, independent of the molybdenum binding site, where where oxygen gets reduced at FAD. So that kind of makes sense. It can also reduce N-oxides. I feel like that could also be a drug hunter topic. Some people love them, some people hate them. I am neutral. And perhaps one that we may not be looking for is that it can actually ring open heterocycles, like the benzoisothiazole in this example of ziprasidone. The one last one one last class of reactivity that I want to note is that it can actually do amide hydrolysis. And this was something that Genentech came across with their example of GDC 0854, which is a BTK inhibitor. And what they found in the clinic was rapid metabolism of their compound. And despite it having a completely acceptable preclinical safety profile. So this reference, also the Drug Metabolism Disposition Journal, studied this metabolic pathway, and Genentech has then since further disclosed SAR to obviate the issue. But characterization of this really uncommon AO activity has been done in silico, and it's believed that it simply goes through, like shown on the screen, a strictly molybdenum-4 mechanism occurring at the MoCo active site. It can't rule out a 2-electron pathway, for those of you that like to nerd out over these things. It does remark that it would need something to oxidize, but it could be the heterocycle itself that serves that purpose. So I'll leave you with these last thoughts before I turn it back over to Neil. But when you're encountered with an aldehyde oxidase activity, which I highly recommend screening for early in your hepatocyte models like GSK did here, is that mitigation strategies can be bucketed into 4 responses. So the obvious one is to stop the activity and find a new substrate. That doesn't always work. So, to decrease the rate of the metabolism through structural changes, especially if that susceptible motif is part of your pharmacophore. And then less common is to exploit the reactivity like Fasodil does. So I don't know if you guys knew this, but the actually active drug is the product aldehyde oxidase metabolism. And then lastly is to proceed at risk, but really emphasize that you do want to have that thorough understanding of the translational activity for the metabolic species or for the metabolic profiles across your different species. So Neil's going to finish up over the next few slides.
Thanks, Nikki. So going back to our JAK inhibitors where we left off, we didn't talk about the SAR at the 2 position. And it turns out the SAR at the 2 position is what is needed to abolish the aldehyde oxidase activity. Compounds like 14, 15, and 16 are exemplified here as examples that retain the desired potency and solubility profile. They have poor in vitro clearance and have high SLF solubility, which you'll remember the initial pyrazole at this position did not have. So using the known crystallography information of using known crystallography of aldehyde oxidase, compound 11 was docked. And you can see the molybdenum cofactor here in teal in the back of the in the back. And the quinazoline fits nicely and close to the molybdenum cofactor binding site. And this would explain why substitution at the 6 and 8 positions had no effect on AO activity. However, compound 15 was unable to be docked at all, postulating that the basic substitutions at this 2 position clash with the enzyme. So let's take some time to discuss the medicinal chemistry route to these compounds. In some senses, this is a textbook medchem route, where we access the highlighted trihalogenated intermediate that enables orthogonal functionalization at the hinge, back pocket, and ribose pocket. The back pocket group was prepared using standard chemistry to borylate and oxidize the para-ethylfluorobenzene before brominating and protecting up the phenol as a SAM. Standard neuroborylation conditions delivered A in a modest yield. So key intermediate B was prepared from the amino bromobenzoic acid by selective iodination and reduction to the aldehyde before urea and pocyl were used to form the chloroquinazoline. So that chloroquinazoline B underwent copper-mediated amination, preferentially at the chloro 2 position, which was followed by Suzuki cross-coupling with A to install our preferred back pocket, before a second amination installed the amino alcohol. Final deprotection under acidic conditions formed 16. And thinking about my role now, each of these steps were amenable to parallel and array chemistry such that we could explore a large number of different groups in each of the areas. So comparing our compound 11 with the basic compound 16, we do see an increase in lung exposure, with a good window for lung versus systemic exposure. The poor mouse IV PK was as desired. However, the compound in this series as a whole were very high for plasma and tissue binding. And when that's factored in with the dotted lines, we do not believe that this compound is good enough to drive the desired lung pharmacology, especially consideration of the IC90 threshold, which is in the grey line on these plots. So this was a series that was ultimately deprioritized for other chemical matter, finding a balance of potency, properties, particularly free fraction, and PK lung exposure was not achievable. So what do we want to take away from this? Opportunities for improved efficacious therapies for asthma are still needed. Harnessing the anti-inflammatory markers of JAK inhibition is still a viable strategy. Lung metabolism prediction is really difficult, although recent advancements are leading to a more thorough picture. Medicinal chemists should always caution for aldehyde oxidase metabolism and screen early, as Nikki said. But let the science drive your mitigation strategy. So all that's left for me to do is to thank all those at GSK who were part of this project. Some of them are named here on the slide. Thank Nikki for inviting me to be part of this. Thank you to Drug Hunter for inviting both of us to be part of this. And thank you all for your kind attention.
Awesome. Yeah. Thank you both for like a really educational talk. So yeah, definitely learned a lot about like, the lungs, that where the heart is. We have a smaller lobe there, and all these nice strategies of AO metabolism, some of them really being counterintuitive, as Nikki pointed out, reductions. That's very surprising. We have a lot of audience questions already, and let's go right in. So we have Gavin Hurst asking about, you mentioned different AOs in rodents and dogs. How do you think about AO expression levels from preclinical species to human?
Neil, you want to take that or should I?
Go for it, Nikki.
I could take that one. I think that's a really excellent question. And as far as I can tell, that the relative percentages, or if you will, contributions from the different species, they're just not known. So I think you have to do the experiment. I think you need to actually look at the different hepatocytes for the preclinical species that you're going to take through safety going forward. And just, sometimes, as you saw the examples I gave, it was a surprise going into human, and that the preclinical species didn't show it at all. So I think knowing what your structure looks like, and that it could be susceptible, even if you dock, that docking model is for human aldehyde oxidase 1. and there are different isoforms in different species. So I think just carrying and understand that risk as much as possible. And as a chemistry mind, right, you should be able to be like, hey, that could be a substrate. File that away for a later date if I don't see it early.
Yeah, makes total sense. Yeah, thank you, Nikki. And another question here is, it is interesting that you have a phenol with an alpha fluoro group, a moiety which you would have to worry much more in the context of glucuronidation in oral drug. Are there some design principles for inhaled drugs which you could not get away with for oral drugs?
Yeah, absolutely. I guess we mentioned a couple of times through the presentation, really looking for that high systemic clearance. We want high first pass metabolism. We were talking just before we started around the high percentage of the molecule when inhaled that's swallowed, and then the high percentage that gets into the lung that gets straight into the systemic circulation. And so really we're looking for metabolic hotspots to design into the molecules such that they can be rapidly cleared.
Yeah, makes total sense. Something definitely counterintuitive for an oral drug, right?
Yeah.
I guess I have a question of my own. You introduced this kind of basic amine into kind of the side chain. I wonder, in terms of lung delivery, do you have to worry about HERC, or is that not that much of a problem? Because as you said, once you basically hit the systemic circulation, you get much higher clearance, and the amine is not a challenge anymore. Is HERC a challenge in those kind of cases, or is it not at all to worry about?
Yeah, it is a challenge, and it is screened for early. Sorry, Nikki, I cut over you.
No, I was just saying, like, it's very easy to think that just because you have high clearance, right, that a compound when it hits the stomach, but if it has high volume, so you want to keep watching the volume on these compounds. And so by adding a basic amine, if your compound is distributing through lung tissue, the moment that it passes through the lung, it's going to distribute to your systemic tissues as well. So I think it makes this suggestion, I think it's easier in a way that it's probably not as observed of an issue, but it is something we still watch out for, particularly for compounds that are having a higher lung volume tissue distribution.
Yeah, makes sense. Cool. And then Sandeep is wondering about, given the variability in aldehyde oxidase activity across species and the challenge in predicting human PK, what strategies do you recommend for early screening to mitigate AO-related liabilities in drug development? So you talked about that a little bit, Nikki, but like maybe if you could elaborate on a few maybe assays or other strategies, that would be, I guess, helpful for Sandeep.
Neil, do you want to pick that up with the GSK screening strategy that we alluded to?
Yeah, sure. So we, as mentioned in the talk, used human liver cytosol plus and minus an aldehyde oxidase inhibitor to try and pick apart that the component of that metabolism that was due to the aldehyde oxidase. And obviously, we went beyond that to lung homogenate to see what component of that was specific to the lung tissue. I think using a panel of different metabolic enzyme inhibitors through your microsomes and hepatocyte assays allows you to start to pick apart that clearance and understand which enzymes are contributing most.
Yeah, that makes a lot of sense. Nikki, you want to add anything or?
Nope.
No. Okay. Sounds good.
For once.
In terms of the chemistry, Ramil is wondering, I never heard about a metal catalyzed cross-coupling being selective for chloro versus bromo or iodo. Is this not an SNAR reaction? Could you comment on that reactivity? Maybe we can go back to the screen. I probably have missed that.
Yeah. Get the right screen.
Perfect.
Yeah, so you clearly would expect the SNAR to go at that 2 position first. My recollection is that we saw probably what is an SNAR mechanism under those conditions, heating up. But the highest yields in those transformations were under copper mediated conditions. At the time, we did quite a lot of screening for different ligands for the copper and different bases to identify conditions that were selected for each of the positions.
Yep, makes sense. Cool. Yeah, thanks for the comment on that, Neil. And then I guess we have a question maybe also for Neil, given, as you talked about in the beginning, that those small molecule JAK inhibitors would be intracellularly. Muhammad is surprised to see you chose the compound not to penetrate cells well based on the properties. Could you comment on that, like how cell permeable the compound is? If you kind of limit permeability, I guess, we have to define what permeability we mean here, like permeability into other tissues or cell permeability. So I think Mahamud's question is wondering about that.
Yeah, so I guess as part of our screening cascade, we look at permeability. We very early in our assay cascade had a number of cellular assays where we confirmed that we were getting sufficient cellular concentration for activity. And also at GSK, we use a cellular concentration assay to understand how much of our compound is getting into the cells. I guess we're not looking for sort of classic absorption permeability from the gut in this inhaled space. So put a lot more focus on the cellular assays and the cellular concentration as opposed to classical permeability assays.
Cool. Yeah, those are very different than KCO2 assays and MDCK assays, the kind of intracellular concentration assays, right? So it's important, I guess, to understand those differences. Yeah, thanks for elaborating on that, Neil. And then we have another question from Indrawan. Did you worry about varying exposures in upper airways versus the rest of the lungs? And how can you test that? Nikki.
Yeah, I asked that you ask, because I think that's a fantastic question that we kind of glossed over. And in a human, you want to aerosolize it, right? And we can do modeling studies based on aerolization and how far that will penetrate. But really, the way to do this preclinically is imaging. And so if you're going into your very early studies, I would say what we did with mice and like intranasal or intratracheal dosing is that you can actually look at different tissue concentrations and see where it is being distributed and by how much. I think the caveat to that is that there's a lot of variability. So in intranasal dosing, they're putting solution, like a little drop right at the edge of the mouse nose, and he inhales it, or she. And how that happens, a lot of this will also so how that happens can be very variable, particularly if it's not a solution. When it's a solution, it's usually the standard way to go. I was saying a rat, one of the things you can do is intratracheal dosing. And that's where you take basically and you dose it down right at the top where you bifurcate into your esophagus bifurcates to your trachea, to your lungs, and to your stomach. Keep in mind that for an inhaled dose, half of that in a human ends up going to your stomach to begin with. And so that's why we also look for poor oral properties as well. But what I found was fascinating in a rat is depending on who administered it and what angle the rat was held at, it would deposit into different areas of the lung. And that makes it really difficult. I mean, gravity played a role in this. So that makes it very difficult for consistency from experiment to experiment. But when it comes to the higher studies as well, and even in rodents, you can do imaging if you really want to understand that as well without something as crude as like tissue concentration.
Yeah, no, super cool. Thank you for elaborating on that. That I think clarifies a lot. And yeah, very surprising. Different angle causes different exposures. Yeah, you wouldn't think about that too much. Details matter, as we talked about in the beginning. I think we're almost at time. Maybe one last question I'd like to ask from an anonymous attendee. Is there a particular reason you chose to go for a pan-JAK inhibitor? Because even if your compound seems to have lower blood concentrations, there could still be an amount that goes to the blood, which could cause on-target toxicity. Neil, you talked about the neutropenias and potentially anemias from JAK inhibitors. We know that they're used for myelofibrosis, for example, a lot. So maybe could you comment on the strategy that you were going for a pan-JAK inhibitor, also maybe hitting TYK2 as well.
Yeah, so across different projects at different times and different chemical series, we have looked at, I guess, more than aiming for specific selectivity, looking for sparing certain JAKs, like avoiding TYK2 activity and aiming for JAK 123 or JAK 13, or there's different selectivity profiles that we have explored at different times. Aiming for a pan-JAK inhibitor really was driving up that full response, and trying to understand whether we could get sufficient window through the inhaled route. But as have other companies, we also looked at different selective options.
That's good. Cool. Yeah, I think that wraps it up very nicely. Thank you both, Nikki and Neil. This was a phenomenal talk. Really learned a lot, and I guess we had a lot of audience questions too. So thank you to the audience for an engaging discussion in this. So for me, I'm left with thanking everybody for the attendance today. And we are actually going on a little spring break here at Drug Hunter Flash Talks. The next Flash Talk will be on April 17th. And we'll be talking to Ulrich Lücking about sulfoxamines in medicinal chemistry. So an emerging functional group that I would invite everybody to, again, on April 17th. And with that, I wish everybody a wonderful day. Again, thanks, Nikki and Neil, for coming today and presenting on this really interesting topic. And, so many counterintuitive learnings I had. And again, you proposed a few nice articles we could write up on Drug Hunter, which I will follow up on. And with that, have a wonderful day, everyone. And see you next time.
Thanks for having us. Bye, everyone. Thanks for joining.
Bye-bye.